Good morning, everyone. So today we will continue with chapter 3, looking at how our cells are using energy, how our cells are processing energy um, in biochemical reactions. I want to remind everyone that we're going to be thinking about this conceptually. You don't need to worry about equilibrium constants, enzyme kinetics. Don't worry about any of the formulas that are in this chapter. I want you to just be thinking conceptually, how does a cell manage all of its resources, all of its matter resources, all of its energy resources? And in that context, we're looking at different types of uh, chemical reactions as we left off on Monday, looking at um, spontaneous versus non-spontaneous reactions, looking at different types of chemical reactions that either will be what we call energetically favorable, or energetically non-favorable. So let's repeat that slide here for just a minute. Here we have an example of molecule Y being converted to molecule X, and we're saying Y has more energy. And when we talk about the free energy of a molecule, we're really thinking about the chemical energy, which is potential energy, the chemical energy in its chemical bonds, right? The electrons that are holding together those atoms. So in this case, this is an energetically favorable reaction because the conversion of Y to X will increase the disorder of the universe. Right? Will be releasing energy that may be adding to the disorder, the most disordered or the lowest form of energy, as in thermal energy. But that process, that chemical reaction can occur spontaneously, so we say that it's energetically favorable. The opposite reaction, in which X is converted to Y, a lower free energy molecule X converted to the higher free energy molecule Y, will require energy to be put into that, right? That's going to consume energy. It requires energy. So we call that an energetically unfavorable reaction. And that reaction of X to Y, this non-spontaneous reaction, can only occur if it's coupled to a second reaction that is energetically favorable. And so we will be talking about some of these types of energy requiring reactions and how it is that they can be driven forward by being coupled to a second spontaneous energetically favorable reaction. So let's look at these types of spontaneous versus non-spontaneous reactions in sort of a different way. Here we're looking at an energy profile of a reaction of Y, reactant Y, being converted to product X. If we look at the progress of this reaction from left to right, we see that Y, this is a spontaneous reaction because the free energy in molecule Y is greater than the free energy in product X. So we say that the overall change in free energy, delta G, is negative, right? This is spontaneous. Overall, energy will be released in this process. Now, what we also see is that Y is not always converted to X because this chemical reaction has to overcome this energy barrier that we call the activation energy. And that activation energy can be supplied by a spark, by an energetic molecular collision, by the raising the temperature of the reaction. But usually in biological situations, cells can't um, the energy or the heat energy that would be required to convert Y to X or overcome that energy barrier is greater than what would end up destroying cells. So, and we'll come back to that a little bit later. But a spontaneous reaction is converting higher free energy molecule to or compound to a smaller to a molecule or a product of lower free energy. So overall, this is exergonic. Overall, energy is released in this process. And that energy from a spontaneous reaction could be captured and transferred to a free energy molecule like ATP that could then be used to do other work in the cell. The opposite type of reaction is non-spontaneous. We call this endergonic. If we look at the energy profile of an endergonic reaction, we see that the reactant has lower free energy than the final product. So it has a change in free energy that is greater than zero. It's positive. Energy must be supplied for this reaction to be able to occur. And again, that energy that must be supplied may come from hydrolysis of ATP. It may come from the light energy of sunlight. Again, this reactant, even when it's converted to Y, requires even more energy to be supplied than would be required. The difference between X and Y still has to overcome this energy barrier, this activation energy. So we'll be looking at those types of reactions, and we'll look at how or what type of energy can be supplied to drive the conversion of a reactant that has lower free energy as it's being converted to a product of higher free energy. And this type of non-spontaneous reaction is what we're talking about when we describe the formation of organized, complex, higher order molecules. For example, the formation of a polymer. The reaction that will join subunit A and subunit B, that condensation reaction, is energetically unfavorable. It's non-spontaneous. The cell must be able to provide an energy source in order to join two subunits to form a polymer, right? And to add another unit and another unit. So building a more complex, a more ordered molecule is going to require energy, this condensation reaction. While breaking down this complex polymer, that hydrolysis in the presence of water, breaking it down to its constituent subunits is energetically favorable. That's a spontaneous reaction. Energy will be released. Both cases, both types of reactions are actually going to increase disorder of the universe. They're going to follow the second law of thermodynamics. In the case of a condensation, energy will have to be supplied. A coupled reaction that is spontaneous will have to be available in order for a condensation reaction to occur, in order to drive this energetically unfavorable reaction. So we're going to look at examples of condensation reactions, examples of hydrolysis reactions, and look at the source of the energy that is required for condensation, or the energy that is released in a energetically favorable spontaneous reaction. So let's start looking at some specifics, some examples. One type of condensation reaction is a building of a macromolecule. And one type of macromolecule in our cells is glycogen, a polysaccharide, a polymer of sugar subunits. The subunits are glucose molecules. So this reaction of building glycogen, this higher ordered, higher energy molecule, is going to require energy. Energy has to be supplied in order for glucose subunits to be joined together to form that glycogen molecule. And so we would say that glycogen is a higher energy molecule than glucose is. And again, conceptually, because energy was consumed, energy was required in order to join glucose subunits together to make this polymer, then glycogen is a higher energy molecule. So let's use glycogen as a higher free energy molecule compared to a relatively lower free energy glucose molecule as examples when we look at reactions and the favored direction of a chemical reaction. In this example here, where we have Y and X. Y can be converted to X. We're going to say that Y is the higher free energy molecule, glycogen. X is the lower free molecule, glucose. And the favored direction of this, energetically favored direction of this reaction is from Y to X, right? There would be the delta G, the change in free energy, delta G would be negative, less than 0, as glycogen is converted to glucose, right? That heavy arrow is showing that that's a spontaneous reaction. But of course, because there's always some random motion of molecules and atoms, there is always some thermal energy. Some X will always be, rarely, but some X can be converted back to Y. Right? Chemical reactions can occur in both directions, but the favored reaction is the spontaneous direction, in this case from Y to X. So if we keep looking at thinking of Y as the higher free energy molecule glycogen and X as the lower free energy molecule glucose, if we start with an equal number of both Y and X in a solution, what we will find is that most frequently Y, or very rare, frequently, Y will be converted to X, right? That's going to occur most often. But every once in a while, X will be converted to Y, right? Because these chemical reactions can occur in either direction. But the favored direction is from high free energy molecule Y, which would, in our case, would be glycogen, lower free energy molecule X, which in our case would be glucose. And therefore, because more frequently Y is converted to X, relatively rarely X is converted to Y, we're going to end up with a increase in the amount of X to Y. So the ratio of X to Y is going to increase as this reaction is moving towards equilibrium, which again, all processes, all spontaneous processes are always moving towards equilibrium. Now remember what we mean by equilibrium. Equilibrium is not a state in which there is an equal amount of reactants and products, but equilibrium is a state in which there's no net change in the amount of reactants and products. So every time Y is converted to X and X is converted to Y, at that point, that reaction has reached equilibrium. Now, that's also something important for us to think about, even in the context of what we've talked about before. If we think about diffusion, diffusion is a spontaneous process where solutes are moving from a region of high concentration to low concentration, right? They're moving towards equilibrium. And the only way that a cell can prevent the concentration of solutes to be equal on both sides, for the solute to reach a concentration equilibrium, is to use energy in the form of ATP to pump, for example, sodium ions out of the cell. So keeping a reaction or keeping a process from reaching equilibrium is going to require energy, either in the case of a chemical reaction, either adding excess reactant or by removing product. So in the case of what we're just looking at here, if we allow the reaction X and Y to to keep converting from one to the other. Eventually, we will have an excess of X because more Y is converted to X. Fewer times was X converted to Y, but eventually the reaction reaches equilibrium so that every time a Y is converted to X, an X is converted to Y. Right? So we end up with the same rate of the reaction in both directions. Neither direction is favored. And at this point, when we're at equilibrium, the change in free energy is zero. Right? The same amount of free energy as on either side of this reaction. Now, we can also then ask, well, what if we, instead of starting out with an equal amount of X and Y at the very beginning of our experiment, what if the starting amount of X was much higher than the starting amount of Y? Which direction of that reaction would have been favored initially, right? At the beginning of a reaction where you had an excess of X relative to Y, what would be the overall reaction? Or what would be the overall favored direction of that reaction? Anyone like to try?
Would it be to the left?
It would be from which to which?
From X to Y?
From X to Y, yes. So this is reminding us that not only is the change in free energy of a reaction or the favored direction of a reaction is not only due to the difference in free energy of the individual molecule. So Y, if the Y is our glycogen, glycogen has more energy, more chemical energy in its bonds than X does. But if we have more X or glucose, the favored direction would be from glucose to glycogen. So when we're considering the free energy change in a reaction, that's reflecting the degree to which that reaction creates a more disordered state of the universe. So we have to consider not only the difference in free energy between the two components, the products and the reactants, but also the relative concentrations of those products and reactants. So as we just said, even though Y to X is the favored direction of this reaction because Y has greater free energy than X does as individual molecules, if there is a great increase in the amount of X, then the reverse reaction would be favored. So if we think of it in the context of our, we can even go back here for a second. If we think in the context of Y is glycogen, X is glucose, if our cell has a huge amount of glucose, if glucose keeps being transported into the cell, our cells will favor the chemical, biochemical pathway of converting glucose molecules into the polymer glycogen, right? Even though that on its own, the individual steps would be, requires energy, right? The large excess of glucose in the cell would favor the formation of the more complex, higher energy molecule glycogen. So not only the difference in free energy of the molecules, individual components, but the ratio of the concentration of each of those as well. So this is important when we think of our metabolism, which of course is a interconnected web of huge numbers of different biochemical pathways. So individual steps in these different pathways are joined with each other. A product of one reaction is the substrate of another reaction. So what we can do is then let's put some of these reactions together, because in reality, in a cell, we don't have just one chemical reaction going on. We have sequential reactions and interdependent reactions. So let's consider two different reactions. X converted to Y. Now, X is lower free energy, Y has higher free energy, because at the equilibrium point, we would have more X than Y. So X to Y is the unfavored direction of the reaction. But let's say we also have a second reaction, and that Y is converted to Z. This reaction is favored in the forward direction. The spontaneous direction of this reaction is higher free energy Y to lower free energy Z. At equilibrium, we'll always see more Z than Y. And then we can put the two of those together. So we have X to Y is non-favored, Y to Z is favored, energetically favored. But what if we put them together in a sequence of reactions? I'm using our arrow to show the degree of how much the reaction is favored. So for example, Y to X would have a negative delta G, but Y to Z has a negative delta G, but it would be a bigger difference, a bigger change in free energy. So we have this larger arrow indicating that Y to Z is greatly favored. That's more favored than Y to X is. So in both of these reactions, Y is the higher free energy molecule, because if we look at equilibrium, for every molecule of Y, there will be more X. For every molecule of Y, there will be much more Z. So then the question is, while Y to X is favored and Y to Z is favored, if we put them together in sequence, what would be the overall direction of this reaction sequence if we started with, now this is equilibrium, but if we started with equal amounts of X and Y and Z? What would be the overall direction? Would the overall direction be from left to right or from right to left?
Left to right.
It would be from X to Y to Z. Right? Remembering that some X can always be converted to Y, though the favored direction of this reaction all on its own is Y to X. But every time a Y is available, it will be greatly favored to be converted to Z. So we would end up at equilibrium having more Z than the other components at equilibrium. We'd always have the least amount of Y. And so what we're showing is that even though X to Y is unfavored, the fact that Y to Z is very favored, the overall reaction will be from X to Y to Z. And this is actually more close of an example of what goes on in our cells. We could start, and we will be talking about in our next chapter, glycolysis, the breakdown of glucose. Now, glucose is a molecule that has some energy, free energy. It has to be converted to a higher energy molecule like glyceraldehyde 3-phosphate. That's an unfavored reaction. ATP has to be added to that process in order for glucose to be converted to G3P. But the G3P being converted to pyruvate, the lowest energy molecule of this reaction, is highly favored. So overall, glucose is converted to pyruvate in glycolysis, even though in that multi-step process, some individual conversions are favored and other individual conversions are not favored. Right? So that's what we're getting ourselves ready to be thinking about. In glycolysis, glucose is converted to pyruvate, the lower energy molecule in this multi-step process. Sequence of reactions. Some are favored, some are not favored, but overall, the sequence is favored because overall in those 10 steps, the high energy molecule glucose has been converted to the lower energy pyruvate. And along the way, some energy is released, transferred, and essentially held in ATP. So that ATP can do work, other types of work in the cell. And that's really what our metabolism is, this web of sequential reactions. Living cells are able to allow these different reactions to keep moving forward, even if some individual steps are unfavored, as long as the end products of metabolism are either released from the cell, like carbon dioxide is released from the cell, or the end products are consumed, that they are fed into another reaction. And as long as there's a constant input of new substrate, all of these reactions will be moving forward. So the only reason that these reactions can work in a living cell is because a living cell is an open system. A constant input of substrate, a constant output of products allows these sequential reactions to keep moving forward. So I have a question for you. What if all of those different biochemical pathways reached equilibrium? Instead of some of the pathways being favored and some being unfavored, they're all moving towards equilibrium. And what we're asking is what would happen if all of those reactions reached equilibrium? All right, how's everybody doing? About 5 more seconds. All right, let me close it up. If all of those reactions reached equilibrium, the cell would not be able to survive. If we look at the example we've just been following, if glucose in its glycolysis pathway, all of those reactions reached equilibrium and all of the glucose, or most of it actually, was converted to pyruvate, then in the absence of any more substrate coming into the cell or any product leaving the cell would be dead. And the cell would be dead for a number of reasons, one of which is that the cell would not be able to generate any more ATP. The cell would not be able to extract any of the energy in that high energy molecule glucose and transfer that energy to ATP. Without the ability to be able to generate ATP, the cell would not be able to do any work. It would not be able to transport substances against concentration gradients. It would not be able to drive forward any non-spontaneous chemical reactions. It would not be able to do any mechanical work. So while all spontaneous reactions are moving towards equilibrium, in fact, all reactions are moving towards equilibrium, if they actually reached equilibrium, then the cell would be dead. So let's go back to some of these individual reactions. For example, one type of reaction is spontaneous. Y has higher free energy than the product X does. The conversion of Y to X releases energy. We say it's spontaneous because it will be adding to the disorder of the universe. But being spontaneous doesn't mean that it happens quickly, and spontaneous doesn't means that it may not even occur at all. And the reason for that is this conversion of Y to X requires Y to get over this energy barrier. To get to a more a state that is more unstable, has greater free energy, that a state in which it can be converted to X. So Y actually has to change to get to a distorted, a higher unstable state in order for it to be converted to X. And so let's look at an example of a spontaneous reaction. A simple spontaneous reaction, exergonic, energetically favored, is the conversion of the disaccharide sucrose to its monomer components, glucose and fructose. In order for that reaction to occur, and it's spontaneous with a negative delta G of 5.5, this reaction, the sucrose molecule, has to go through a high energy intermediate, an unstable intermediate. The glycosidic bond that is joining those two monomers has to be distorted in order for that glycosidic bond to actually break and form new bonds that results in the release of glucose and fructose. That reaction is going to take forever. Sucrose in a beaker is not going to break down to glucose and fructose. It would take hundreds, if not thousands of years for a random event to break that bond. But if we add an enzyme specific for distorting that glycosidic bond, and we call it glycosidase or sucrase as a specific example, then the entire solution of sucrose will be converted to those monomers immediately. So in a cell, cells are regulating their chemical reactions by regulating the activity of enzymes, an enzyme that will accelerate a specific chemical reaction according to the cell's needs. So what is the enzyme doing? The enzyme is lowering the activation energy, the amount of energy that the reaction needs in order to proceed. Reactant Y needs to be converted to this unstable intermediate state in order for Y to be converted to X. In the absence of an enzyme, it may take a while for that to be able to occur. Sure, heat could be added to the system. The energy could be heat energy, and that could happen in a beaker. But in a cell, heating up a cell in order to drive a particular chemical reaction would probably kill the cell. It would cause denaturation of all of the other structures in the cell. So what a cell will do is activate a particular enzyme, and that enzyme will lower the activation energy, will lower the amount of energy that must be added to the system in order for the reactant to be converted to product. And therefore, the rate of that reaction will be increased. So what are the kinds of things that an enzyme does? An enzyme is a protein that has a particular three-dimensional shape. It has an active site, the site at which it can actually bind this substrate, the reactant. And what it might do, depending on the enzyme, depending on the substrate, what reaction it's catalyzing, it may end up actually putting contorting the bonds of that substrate. It may orient two different substrates relative to each other such that they can form new bonds between each other to form a product. It may actually add a pocket of acidic amino acids and transfer a proton to the substrate in order to convert it to a product. Different types of enzymes will interact with a substrate in different ways in order to help or accelerate the conversion of the reactant to the product. Right? Again, specific reactant converted to specific product, the cell is going to direct the needs of the cell, catalyzing, accelerating the rate of specific reactions, right? Allowing that reaction to occur rather than heating up the cell, allowing all reactions to occur and heating it up to such a point that the cell may actually have all of its structures denatured, degraded, causing the death of the cell. Now, the other thing that an enzyme does, it's increasing the rate of the reaction. But remember, chemical reactions can go in either direction. And so if we look at the reaction of X to Y, and in this case, Y is the lower free energy molecule, in an uncatalyzed reaction, a reaction in the absence of an enzyme, at equilibrium, every molecule of X that's converted to Y will also have a molecule of Y being converted to X, right? So the rate of the conversion in both directions is the same at equilibrium. If you add enzyme to that reaction, you will still have the same relative ratio of X to Y, the same equilibrium point. It just means that Y converted to X is faster and X converted to Y is faster and Y converted to X is faster. The rate of those reactions will be increased, but the relative ratio of those reactants at equilibrium will not be changed. So that reminds us also that while enzymes are specific, enzymes are going to accelerate or accelerate the rate, catalyze chemical transformations. Enzymes can function in both directions. The enzyme's not altered. That's why it's a biological catalyst. But enzymes can convert the substrate to product and product to substrate, both directions. But if the difference in free energy between the substrate and the product is very large, for example, the product has much lower free energy than the substrate does, then the enzyme is going to catalyze the forward reaction exclusively. Right? And that's what we're talking about in the example that we just showed above, right? This spontaneous reaction, right? Adding enzyme is going to accelerate this spontaneous reaction, the conversion of Y to X in that exergonic reaction, spontaneous reaction. But what about a non-spontaneous reaction? What about an endergonic reaction? In a reaction of X to Y, where Y is higher free energy, what will happen if you add enzyme to that? Essentially, you add enzyme to a beaker of X, what's going to happen? So think about the function of enzymes in a cell. What's an enzyme going to do to an endergonic reaction, a non-spontaneous reaction? About 5 more seconds. All right, let me close it up. And this forward reaction will still occur as long as energy is available to the reaction. What we're trying to illustrate here is the enzyme is going to speed up a reaction, but it's not going to change the equilibrium point. It's not going to replace the requirement for energy, right? And energy is still going to be required in order for X to be converted to Y. So if we look at this endergonic reaction here, the rate at which X is converted to Y can still be increased in the presence of an enzyme. That presence of the enzyme is not going to replace the requirement for energy in order to convert X to Y. All that will happen in the presence of the enzyme is that this barrier, this additional energy, activation energy, will be reduced. But the difference in free energy between X and Y, this amount of energy still needs to be supplied to the reaction. So what is that energy source that is used most frequently in a cell to help drive this conversion of a lower free energy molecule to a higher free energy molecule? What is the energy source that will help drive a spontaneous reaction? And that's our ATP. We call ATP an energy carrier. It's a source of useful energy that's available in the cell that can be used to drive non-spontaneous reactions. And the reason is that ATP can be hydrolyzed. It is a highly spontaneous reaction, conversion of ATP to ADP. That is an energetically favored spontaneous reaction. And if ATP hydrolysis is coupled to a non-spontaneous reaction, it can be used to drive that non-spontaneous reaction forward. So let's remind ourselves the difference between ATP and ADP. ATP is our high energy, mobile energy carrier. ATP is formed by adding a phosphate molecule, a phosphate functional group, to an ADP. So ADP is a lower energy molecule, ATP is higher energy molecule. The energy that is required to form ATP comes from light energy in sunlight, or is extracted from high energy food molecules. That ATP is formed as our ribose nucleotide with an adenine, 3 phosphate groups. That's our adenosine triphosphate, right? 3 phosphates. Those phosphates are linked to each other by high energy phosphoanhydride bonds. High energy meaning energy is required in order for those bonds to form. And if we look at our ATP, one way to think of it as to why it is a high energy molecule is you look at all of those negative charges on all of those phosphates. Those are repelling each other. And so this triphosphate molecule is sort of like a compressed spring. All of those negative charges repelling each other are ready to be released. And so ATP in an aqueous solution in the presence of water can be hydrolyzed. And the hydrolysis of ATP, the cleavage of that terminal phosphate, converting ATP to ADP plus an inorganic phosphate, releases energy that is now available for the cell to do the work of being alive. ATP hydrolysis can drive transport. ATP hydrolysis can drive mechanical work like cells dividing, chromosomes moving, vesicles walking along the cytoskeleton. And ATP hydrolysis can drive chemical work of the cell. And that's what we can see on the next slide. If the cell needs a reaction, X converted to Y, this non-spontaneous reaction, right? X is lower free energy, Y is higher free energy. How does the cell drive this favored, this energetically unfavorable reaction. If that reaction is coupled to ATP hydrolysis, conversion of high free energy ATP to lower free energy ADP that has a large negative delta G, like a set of gears, ATP hydrolysis can drive the non-spontaneous conversion of X to Y. The reason is negative delta G, more energy is released in conversion of ATP to ADP than conversion of X to Y needs. As long as the positive delta G is less than the negative delta G of ATP hydrolysis, we can couple that hydrolysis to the driving forward of this non-spontaneous reaction. And that's what we're going to talk about in the next set of slides. I'm going to wait until Friday to start that, but what we will be looking at are several examples of coupling an unfavorable, energetically unfavorable reaction with the energetically favorable hydrolysis of ATP to produce an overall favorable reaction in being able to convert, for example, X to Y. So we'll stop here. We'll pick up on this page on Friday, and then we'll start looking at examples as we think about the types of chemical reactions that we will be describing in our next chapter, some favorable, some unfavorable, energetically unfavorable reactions in the set of reactions of glycolysis, followed by the rest of aerobic respiration in our next chapter. So we're thinking about energy processing, thinking about requirements for energy as a cell manages its matter resources, manages its energy resources, getting ready to thinking about the energy generating organelles of our cell. All right, so we'll stop here. Hopefully everybody has a chance to read through the material, get yourself comfortable with this, and then we'll be ready for looking at cell respiration in our next chapter next week. All right, see you later. I also have office hours today at 1. If anybody has questions, I'll be in Zoom at 1 o'clock. See you later.